In today's session, we will be examining the fungi and we will talk about their classification, their properties, and how you might diagnose them in the lab. Let's start by defining the fungi, just like we did with viruses, with prions, with vaccines. So fungi are eukaryotic. They do have a nuclear membrane. They are heterotrophic organisms as opposed to autotrophs. So autotrophs like plants, they can make their own food substances, their own macromolecules, as opposed to fungi, which usually do not, and they obtain them from external sources. They can obtain these nutrients by absorption. We'll talk a little bit about how they do this later on. They produce non-motile spores for the most part, but yeah, this is with exceptions. So this definition does not account for some of the motile spores, and we'll talk about these. The motile spores are not medically relevant. We will talk about these in a little bit. So these spores are produced on mycelia or yeast thalli, and we will define what these means in a little bit. But for now, what you should know is that the fungi can be of the yeast form or what we call the mold form, and the mold usually grow in what we call mycelia. We will be further exploring this on a upcoming slide. Um, these organisms, they have cell walls, and these cell walls are mostly composed of chitins in their assimilative stage. Assimilative means vegetative state. This is the state during which they can basically obtain nutrients, and they are growing in that state. They're not what we call spores in that state. So in their assimilative stage, when they are not spore-like, or when they are actually obtaining nutrients and growing, they have cell walls that are composed of chitin. Let's examine some of their characteristics, further characteristics. So they are eukaryotic, like we said, because, well, they do have a nuclear membrane or nuclear envelope. It is there. They may or may not have mitochondria, but they definitely do not have chloroplasts. Chloroplasts are found in plants, and fungi are not plants, so they do not have chloroplasts. Their cell walls have one of the major components of the cell wall is chitin, uh, but they may have other non-cellulose polysaccharides. They do have glucans, for example, and rarely you will find cellulose. And frankly, the ones that do have cellulose in their cell wall, these are not medically relevant to us. As for their nutrition, they do absorb nutrients from the outside. Some of them produce enzymes to break up these macromolecules and then absorb simpler nutrients, and some of them just can only rely on simple nutrients for absorption. Because, well, they are heterotrophs. They are not autotrophs. They do not make their own energy-based macromolecules for consumption. Um, as for motility, they are mostly non-motile, with some exceptions. We'll talk about these. Multicellularity is present in most forms, but again, we'll redefine multicellularity as you'll see in a little bit. Um, they don't have a nervous system, of course. They don't have centrioles, and well, centrioles are one of the major components are the centrosomes. We usually use centrioles for our cell division. Fungi do not have them. They have something that resembles centrioles that are called polar bodies that are formed for usage during cell division, but they don't have centrioles like our cells do. As for ploidy, they're mostly haploid, so they are 1N organisms. For the most part, they don't have a duplicate set of their genome within the same cell to call that diploid, as we have in most of our cells. It's higher eukaryotes. Um, and then polyploidy is also possible, and again, we'll talk about this in a little bit. You'll see why polyploidy is possible. Cell membrane sterol is mostly ergosterol, and so what is our own major sterol? What do we mostly have as sterol in our own cell membranes? You answered cholesterol, you're right, and this is one thing that we might be able to exploit pharmacological purposes if you want to treat a fungal infection. So remember, when we try to treat organisms or infectious agents, we try to exploit differences between our biologies. And so we have to bear in mind that these are eukaryotic organisms. They are not higher eukaryotes, but still we have a lot of similarities between us and them. And so treating fungal infections might be difficult because of how similar we are, how many similar pathways we have. So ergosterol versus cholesterol, so it's a different major sterol, and you'll see later on during the antifungal agents lecture that we do actually exploit the fact that we have uh, different types of sterols. Um, as for respiration, they are facultative and some of them are obligate. Aerobes, they cannot live in the absence of oxygen. As for saprophytism, most of them are saprophytes, and this means that they rely on decaying agents, non-living uh, agents out there in the environment. They don't need a living host, they don't need living tissues, but some of them do require a living host. But most of them are saprophytes that live on dead or decaying matter. As for propagation, they can form sexual and asexual spores, and we'll go into these a little bit later during this lecture today. We have two major types of fungal thalli, and thalli stems from the singular thallus, and usually a thallus is a vegetative body that doesn't have structured form. It doesn't have organs, for example. So it's a vegetative, non-structured body. So when it comes to fungi, we have what we call the yeast, and yeast are usually unicellular, and when you grow them, when you culture them on a plate, their colonies resemble those of bacteria. For most of them, they resemble bacterial colonies with some differences, of course. Um, as for mold, these grow in these filamentous forms that we call hyphae, and we'll further explore these in a bit. And they have funky looking structures. We'll talk about these as well. So mold grow mostly as in the filamentous form, which we call hyphae. And as for their colonies, again, they are also funky looking. They have pigments, they are large, sometimes smaller. They might have one color on one side of the plate, but then if you flip the plate around, there's a different color there. And they sometimes have textured colonies that are fuzzy, for example, or velvet-like. So these are major differences between mold and yeast. And then we have organisms that are dimorphic. So some organisms are true yeast, some fungi are true yeast that can only grow in the yeast form, and some of them are true mold that can only grow as mold in their filamentous form. And some are dimorphic and they can switch between these two forms based on environmental cues. And for the most part, this is temperature. And again, something we'll talk more about a little bit later down the road. All right, so take a moment to think about this question. Um, So what is a pertinent difference between fungal cells and human cells? Is that A, presence of ergosterol in most fungal cell walls, B, absence of a nuclear envelope in most fungal cells, C, absence of centrosomes in most fungal cells, and D, presence of chloroplasts in most fungal cells. All right, so if you've answered A, presence of ergosterol in most fungal cell walls, you're wrong because it's present in the cell membrane, not the cell wall. Absence of nuclear envelope in most fungal cells, well, this is also wrong because they do have nuclear envelopes. C, absence of centrosomes in most fungal cells, and yes, this is the correct answer. That leaves us with D, presence of chloroplasts in most fungal cells. Well, definitely not because they are not plants. Let's examine the various forms of fungal thalli. The first form, the yeast, the unicellular forms. Um, these reproduce asexually either by binary fission or by budding. Binary fission means equal splitting of genomic material and cytoplasmic material between the mother cell and the daughter cell, just like bacteria do. Whereas budding means unequal splitting of this material. So we have equal splitting of genomic material. However, there is unequal splitting of cytoplasmic material. So if you take a look here, for example, this is asexual reproduction by budding, and you'll see that the daughter cell is smaller in size than the mother cell. The genomic material has been equally split between the two, but not the cytoplasmic material. As for sexual reproduction, they reproduce sexually by ascospore formation, and this is what yeast asci look like. You see a sac that contains four of these ascospores. As for mold, they grow in the form of what we call hyphae with a singular hypha. A bunch of hyphae is usually called a mycelium or a mat. This is uh, um, what we call, for example, a colony on a plate. That bunch of hyphae is called a mycelium or a mat. The hyphae can either be septate, having these cross walls that separate these filaments into different compartments, as opposed to the aseptate. They don't have these septa, and these are coinocytic, meaning it's one um, tissue behaving as if it's one cell. It's a multinucleated tissue, but behaving as if it's one cell, because there are no cross walls in this case. But effectively, even the septate one are physiologically coinocytic, behaving as if they are one cell, although they have these septa. Why? The answer is on the following slide. So even the septate fungi or the septate mold, they can be considered as if they are coinocytic, and that's because the septa that they have these really gigantic pores that allow the passage of organelles such as a nucleus from one compartment to another. So physiologically, they are rather coinocytic, as if they are one giant cell, despite the presence of these septa. The hyphae might have different forms. They can be hyaline, transparent, that is, because they don't have any pigment. The pigmented ones, like you see on this slide, they are referred to as demataceous because they do have pigment. And these pigments can be of different colors. They can be green, blue, greenish blue, reddish, brown, so on and so forth. And going back to the dimorphic ones or the diphasic fungi, and these, like we said earlier, can switch between the yeast form and the hyphal form, the mold form, and that's mostly based on temperature. They usually grow as yeast at 37 degrees. So this is also the tissue phase because, well, our tissues have a temperature of about 37 degrees. So this is what they look like in our tissues and at 37 degrees in the lab. As for the mold form, usually see it at 25 degrees, which is uh, the room temperature, that is. Fungal reproduction can occur by asexual spore formation or bisexual spore formation. Fungi, yeast, and mold, they can undergo mitosis, they can undergo meiosis, as you'll see. The asexual spores, they are either vegetative or aerial. What do I mean by this? Vegetative spores arise from the vegetative hyphae, and these are hyphae that extend into the growth medium or the growth substrate. If we're looking at plate, this is under the surface of the medium. As for aerial hyphae, they are present on above the growth substrate, above the medium, that is, and aerial spores exist on these aerial hyphae or arise from them. As for sexual spores, they are mostly, for the most part, they are aerial. Asexual spores arise after mitosis, so there's no reduction in the amount of genomic material. As for sexual spores, they arise after meiosis, which indicates there's reduction in the genomic number. So the sexual spores either after meiosis or after fusion of nuclei in sexual cells that or gametogenea, which are specialized hyphae that we call gametogenea. Let's examine the various types of spores. And the reason why I would like you to take a look at the various types of spores and be familiar with them is not just because we want you to remember these names. On one hand, this is relevant because uh, sometimes you have to come up with a rapid presumptive diagnosis, and all you have to do is actually take a sample from your patient and examine it under the microscope. And if you recognize the type of spore you're seeing or the type of hyphae you're seeing, the way it looks, you can figure out what type of organism you have. Let me give you an example of something you will study later on, which is mucormycosis. This is a rapidly invasive type of infection, and you might be able to save somebody's life because this disease has a very high mortality, and you might be able to save somebody's life if you doubt that this is the type of infection. And so what you would do is take a sample, examine it under the microscope, and you will see irregular shaped hyphae. You'll see sporangia, and we'll see what sporangia means in a little bit. You'll see characteristic sporangia of the organisms that can cause mucormycosis, and so you would be able to figure out what treatment to give based on this rapid presumptive diagnosis. And this type of approach has saved many a patient's life, and skilled physician is really one who can uh, try to figure out an infectious agent rapidly in that manner by coordinating, communicating with the lab personnel and figuring out what kind of organism they might be seeing. On the other hand, sometimes you will get questions of this sort on your board exams. If you're doing your MBIME exam, sometimes you do get questions of this sort, they would tell you certain characteristics of spores, and you have to figure out what kind of infection this might be. So again, we're not teaching you for the exam, for the test, uh, but it is a part of it. The bigger concern is for you to keep in mind that you might be able to save somebody's life if you can um, put two and two together and try to recognize these things. Okay, so going back to asexual spores that we also call mitospores because it's asexual. The asexual phase is the anamorphic phase. The vegetative spores, in the past people used to call them thallospores, but you still see that in some references. So the vegetative spores are of three types. First, the blastoconidia or the blastospores. We've seen one form of that, and that's when we have budding from yeast cells. You've already seen that. However, budding doesn't just happen from yeast cells, it can also happen from hyphae, and sometimes you see um, these blastospores or blastoconidia shaped in clusters, like here in this picture. And sometimes you'll see them in chains, sometimes you'll see just one of them arising from the side. So there are different types of blastoconidia that arise by budding, either from yeast or hyphae, and so this is a characteristic of some fungal infections that you will see more and more of as we go along. The second type of vegetative spore, still under asexual spores, chlamydoconidia or chlamydospores. These are actually cells that are already present in the hyphae. Their cell walls become thick, they become enlarged, and you see something of this sort. They can be at the end of the hyphae or the hyphal filament, and we call this terminal chlamydoconidia or terminal chlamydospore. They can be just within the hypha filament, and we call this intercalary, so within. And some of them just branch off from the side. We call these sessile or lateral chlamydoconidia. And these can be present in chains and clusters in different types of forms. And again, some species will, for example, only produce terminal chlamydospores. Some of them will only produce sessile, and then some of them can produce all three forms, for example. The third type are the arthroconidia or the arthrospores oidea as well. These arise after fragmentation of the hyphal filament, and they may have a thickened wall. And then again, we have so many different forms of these. We might have the smooth arthroconidia or arthrospores. You might have the rough ones, which have rough uh, borders. Some of them are hairy, some of them are warty, and some of them just remain in chains along the hyphal filament. So again, it's just um, fragmentation of the hyphal filament that results in the formation of these arthroconidia or arthrospores. The second type of asexual spores are the aerial spores, so the ones you'll find above the surface of the medium. The first one is the sporangiospores, and sporangiospores are usually found within a sac that we call a sporangium, and this sporangium is found on a specialized type of hyphal filament that we call the sporangiophore. Or so this is a stalk that holds the sac containing the sporangiospores. Second type of aerial spores are the conidia, and these come in two forms. The macroconidia, which are usually septate, some of them might be aseptate, but they have these, most cases you see these tapered ends. Sometimes they're not there, but most cases you see these tapered ends. These are the macroconidia. And then we have the microconidia, which are single-celled. So each of these small ones you're seeing here, this broccoli-shaped structure, this is the microconidium holding the microconidia. So the small cells here are the microconidia. Again, the microconidia, you might see them just branching off the side, for example, of the hyphal filament, but they might have some arrangements like clusters, chains, and here we're seeing this rather famous broccoli-shaped structure of microconidia. So still talking about macro and microconidia or conidia in general, they have so many different shapes. As I was saying, the macroconidia, they usually have tapered ends, but you'll see here that these ones do not have tapered ends. They might be long, they might have the broccoli structure, some of them are arising just straight from sides of the hyphal filament. They might be transparent, so hairline, they might be pigmented, so demataceous. And there is a very special form of macroconidia that have cross walls that go in different directions, so both vertically and horizontally. And these are called muriform, so muriform stemming from mural or wall, so they look like a brick wall. So the muriform macroconidia, this is also called dictyospore. The third type of aerial spores is the zoospore, and this is not relevant medically to us. These agents, the chytrids that have these zoospores, they do not infect humans. They are aquatic fungi that may infect amphibians. So the motile asexual spore, the zoospore, is found in a zoosporangium, and they are again not medically relevant to us. Moving on to the sexual spores, and these, like we said earlier, they arise after meiosis or after fusion of nuclei, and these are called meiospores, and this is the teleomorphic phase, the sexual sporulation. So the first type are the zygospores. This is what they look like. This is a dormant spore, just like many other sexual spores. In the most cases, sexual spores arise when you don't have enough nutrients or not enough humidity, and so this is a more preservative kind of condition. The sexual spores, they are more resilient for the most part, and so this is a dormant spore, and whenever you give it enough nutrients, it can germinate and it will form the sporangiospores. You can click here and take a look at the asexual sporangiospores that we had talked about earlier. As for ascospores, and what this is another form of sexual spore, and where In this picture here, I have the mold SI. Uh, they look a lot like villi. And so usually you see eight, in most cases, you see eight of these ASCO spores when it comes to fungi in one ascus. So the singular of asci is ascus. So this villus-like structure is called an ascus, and you'll see eight asci in them. Uh, now, if you go back in the lecture and go to the asci of yeast, yeast usually have four asci in one sac, as opposed to a mold. In most cases, you form but this is not always true, but for the most part. Third type of sexual spore is the basidiospore, and this is, for example, how mushrooms replicate. They are common mushrooms. Um, so here we usually see four of them for basidiospores on basidium. So if you're wondering, so if you take a look, if you're wondering where this exists on a mushroom, if you take the mushroom cap and you examine the bottom, this is what we call the basidiocarp, and this is where the basidiospores are present and they can get released. As for the chytrids, they also can form possibly a type of sexual spore. Again, the chytrids, this is the aquatic fungi that mostly infect amphibians. We don't need to worry about this a lot as human beings um, in terms of human infection. And uh, there were reports of diploid chytrid zygote. This is rarely seen, and actually, it might just be an artifact of uh, bad cell splitting or irregular fusion. And so it looks like a zoospore that has two flagella, so it might be an artifact that doesn't always exist. But some people have reported it. We keep that in mind, but you don't need to worry much about the chytrids or in terms of their relevance to human infection. Rely on the clinical presentation. Some fungal organisms cause characteristic infections or characteristic symptoms. Woods light is a device that's mostly used by dermatologists. This is a UV light that has a Woods filter. What the filter does is basically exclude most of the UV light except for UV light at 366 nanometers of wavelength. And so this is what's transmitted. Some fungal agents, they will fluoresce when they are exposed to UV at 366 nanometers. And these agents are usually skin, nail, and hair infectious agents. And so again, this is mostly used by dermatologists. They apply the Woods light and they will see fluorescence if they have particular types of fungal infections. Radiology and imaging can, of course, be used. As for the wet mount in mycology, you can apply the KOH wet mount, the potassium hydroxide wet mount. In this case, you take scrapings and tissue samples. So for the most part, it is skin, nail, and hair scrapings and tissue samples. It's not deep-seated infection tissue samples. So these scrapings, they are warmed in the presence of 10 to 20% KOH. What this will do is will clear out the organic matter, and so the fungi become more clear. Now you can observe them under the microscope more clearly. So mostly applied for actually keratinized tissues. This is what KOH will clear, mostly keratinized tissues. It's not used for deeper infections. You can also use a stain called the calcofluor white stain along with a wet mount. And calcofluor is a fluorochrome, so it's a fluorescent substance that will bind to chitin in the cell wall of fungi. Of course, so some fungi have cellulose, but that's not the point here. Calcofluor white can bind to cellulose, but for the most part, fungi have chitin in their cell walls, and that can bind to calcofluor white. You can also use tissue stains, and this is, of course, there are multiple types of tissue stains that you can use, and you might enhance the visibility of your organism. And of course, there are serological, immunological, and molecular methods that can be applied in the lab diagnosis of fungal infections. A certain type of stain that can be used in mycology is the India ink or the Chinese ink or nigrosin. This is a negative stain, meaning it will stain the slide. So it doesn't stain the organism itself. So some organisms, some yeast, some fungi, they have very thick capsules that cannot be penetrated. The example here is Cryptococcus, and so these have very thick capsules, cannot be penetrated by the stain. And so you are excluding them from the stain, you're only staining the background. Culture can be performed. Now you have to bear in mind that non-pathogenic fungi and bacteria are fast growers, as opposed to pathogenic ones, which are slow growers. So you want to optimize the conditions so that you can eliminate bacteria and non-pathogenic fungi, which are fast growers. So for the most part, we use Saboros agar, and this agar is usually mixed with chloramphenicol to inhibit bacterial agents and cyclohexamide to inhibit the fast-growing non-pathogenic fungi. And then what you can do is microscopic and macroscopic examination of uh, both a sample from the plate and the plate itself, what the colony looks like. So this will allow you to form or come up with an identification of the organism. And you incubate at both 25 degrees and 37 degrees because you have to catch the yeast, you have to catch the mold, and you have to catch the dimorphic fungi. You want to be able to tell. This will help you in the diagnosis and identification. And usually you incubate for 2 to 4 weeks because these are slow growing organisms. The pathogenic ones are usually slow growers. Take a moment to think about the following question. So what is the major difference between molds and true yeast? Is that A, mold cells secrete enzymes that degrade polymers into absorbable nutrients, while yeast cells do not. B, yeast cells reproduce sexually by ascospore formation, while molds cannot. C, some molds can undergo mitosis and meiosis, while yeast cells can only undergo mitosis. Or D, yeast can produce daughter cells by budding, while molds cannot. All right, so if you're thinking about this question, try to exclude the answers you know are incorrect, and you'll come up with the correct answer, which we haven't explored so far in this lecture. All right, so let's take a look at the different options. Let's start from the bottom up. So D, yeast can produce daughter cells by budding, while molds cannot. As we've seen in this lecture, both types can produce blastocynidia, which are the daughter cells that are formed by budding. C, some molds can undergo mitosis and meiosis, while yeast cells can only undergo mitosis. And if you've been listening carefully when we went over reproduction and then the spore formation, we talked about how both can form uh, uh, basically spores that arise either by mitosis or meiosis. Both of them can undergo mitosis or meiosis, depending on the species. B, yeast cells reproduce sexually by ascospore formation, while molds cannot. Well, ascospores are the sexual spores of both yeast and some yeast, that is, of course, not all yeast do this. So some yeast can do this, and some mold can do this as well. That leaves us out with A, which is the answer. So mold cells secrete enzymes because they are multicellular. They are somewhat more adept at doing things. So mold cells secrete enzymes that do degrade external polymers. So they degrade them outside and then they absorb their nutrients. Yeast cells usually rely on absorbing simple nutrients. As for the classification of fungi, the authority is the International Commission on the Taxonomy of Fungi, so the ICTF. And what the ICTF has come up with as a classification system that was adopted back in 2007 but so far, not a lot of people have adopted this classification system. Around 100,000 species have been identified, and around 100 have been identified as human pathogens. But so you'll see this classification system, you'll see the various types of phyla, of genera, of species, and actually not a lot of people have adopted this because there's a lot of organisms that have not actually been classified, a lot of species that have not been classified into different phyla or different genera based on this system, and so a lot of people have do not really agree with this, and so up to date, people mostly use the older classification system. So this is the older classification system, and I gather you are more familiar with this classification system, which has five phyla, because most physicians and most scientists still talk about diseases and organisms based on this classification system, and mostly based on the phyla. So starting with the first phylum, this is the Zygomycota, and Zygomycota, they get their name from the sexual spore formation, which is zygosporulation, or formation of the zygospores. And if you remember from the lecture, so zygospores, when you give them good conditions, they will sporulate or they will germinate, and you will end up with the asexual sporangiospores. So these organisms, the Zygomycota, they form zygospores, sexual spores. They also form sporangiospores as asexual spores, and different types of Canidia and other types of spores. And one example is Rhizopus stolonifer. This is the black bread mold. So if you've ever seen moldy bread that looks black, not green, if it looks black, this mold is usually Rhizopus, and Rhizopus can be actually pathogenic, and that's something you need to be aware of. Ascomycota, um, the, these form ascospores as sexual spores. They can form different types of other conidia and asexual spores. And one example is Saccharomyces cerevisiae. So this is the yeast that's used for making bread, for making beer, for example. The Basidiomycota, or the club fungi, their sexual spores are the basidiospores, and usually people have not detected really asexual spores for the most part, so it's uncommon to see asexual spores. So they mostly form basidiospores, which are sexual spores. And one example of a basidiomycete or club fungus is the Agaricus bisporus, which is the the mushrooms that we consume, the white button mushrooms. The Chytridiomycota, or the chytrids, these are the aquatic uh, fungi that we talked about that are not medically relevant. They form zoospores as asexual spores, and then possibly the mythic the diploid zygote, which we said may just be an artifact. So this is why we have a question mark. And one example is Batrachochytrium, which has been mostly seen in infections of toads or amphibians. And the last phylum is the Fungi imperfecti, which is the Duetromycota, and they are imperfect fungi, or referred to as imperfect fungi, because sexual sporulation has not been really detected, so it might just be absent. And they can form different types of Canidia or spores. And one example is Candida albicans, and you'll see this is one of the more common fungal infections that can cause opportunistic diseases or opportunistic infections. And you'll see a whole lot more about Candida later on in this course. So this concludes our session, and thank you for listening.